Hi all, I'm Jay, and today we're going to be talking about how the commensal protist Trichomonas musculus exhibits a dynamic life cycle that induces extensive remodeling of the gut microbiota. This was a study done on mice, but of course there's probably going to be a lot of human implication as well. So in terms of why study it, of course the entire premise of this class is how the microbiome affects human health and all other organisms' health, and in terms of how we understand them, protists are generally less understood than others. So the researchers wanted to research how effective can a single protist, like we're going to call it TMU just because it's shorter from now on, be on a mouse's microbiome regarding things like immunity, metabolism, diversification, and bacteria content. And kind of to go along with that, they also wanted to know what exactly is the life cycle of a commensal protist like TMU in the microbiome. So essentially they took mice, inoculated the protist into them, and then they got cecal and fecal samples. Cecal just means the cecum, which is like an intestine part, and then from there they were able to isolate and sequence them, and then they could understand the effects, lifestyle, and other implications of the protist on, stuff like bacterial diversity. In terms of what we already know about protists, we know there's the kingdom of single-celled eukaryotes, and we used to think that they're pretty bad for us. So there's still, studies coming out now that where they find new protists from the ocean and freshwater bodies, and they are really, really bad for us. So we kind of just assumed that was carry over into the microbiome, but now we know, of course, that there are really, really important protists in our bodies that are coming out in new studies like the one by Garrick and Howitt, which basically showed that in terms of immune system stuff, they actually might have a major beneficial impact just by being there. So they could be really, really good for us, and then sometimes, again, they are parasites. They can be bad for us. So we're kind of neutral on protists generally, and we definitely need to learn a lot more, which is why studies like these were made. In terms of the Trichomonas genus specifically, they're usually found in like bovine animals like cattle or feline animals like cats, but they also are found in mice. And then for the specific strain of musculus, we know that they're not inherently pathogenic. So a lot of the studies also kind of going into can it have positive and negative effects as well, depending on what the stress conditions are in the environment. Now, in terms of the importance, once again, potentially the balance of protist, bacteria, and fungus content by itself might have impacts on our immune system. It might not even be that the protist does anything, but it might just be the fact that it exists and takes up space there, that balance is really important for our internal microbiome. But there are also signs that this is also huge for like immune response pathways. So a lot of the times when there's a presence of protists, certain protists, there will be like an activation or a deactivation of certain pathways, right? And then along with that, they inhabit the same spaces as fungi and bacteria, so a lot of people assume that there's going to be some competition, and that's not entirely false according to the study we're about to look at as well. And with the ultimate goal being that this could be translated to humans. So we have Trichomonas, which is a genus that is relative to Trichomonas in mice, so a lot of this stuff might be able to carry over into people, and there are some ethical concerns that I had about this study that might prevent it from being just like actively done on people that we're also going to get into. But yeah, a lot of this stuff might be able to be like extrapolated to people as well. Now, for the overall goal, I just put, how impactful can a single species of protist be on an organism's microbiome. That's the ultimate goal of this. Of course, they're doing it in mice mainly for ethical reasons, but the ultimate goal is going to be to make sure that humans are able to benefit from this research as well. So now we're on to methods. Basically, they took 6 to 8 week old mice, and they basically inoculated them with the sample of TMU, and then at around 12 weeks, they would kind of like take portions out starting then, and then over the course of 28 days, they would collect different samples, and then they'd be able to determine protist content, bacteria content, etc. In terms of the differences, in terms of like the experimental part, they use both wild-type mice, which are just the standard lab mice, and they also use the mutant B-cell deficient mice. So those mice, essentially were like immunodeficient, and then that also has some really cool implications that we're going to get into. And then they also had another difference. So they had germ-free mice, which are basically just, again, they're standard mice. And then they also had conventionalized mice, which are mice that, had some sort of bacterial content in them, and then they received a bacterial suspension to make them, like suitable for kind of like a control group. But they also did have enough of like a prior set immune system that it did also alter results that we're going to see in a little bit as well. So some other random technology that they use: transmission electron microscopy for protein pellet preparation or protist pellet preparation to give to the mice in the first place, and then from there basically they would take the fecal sample and they would centrifuge it, and then that was the way that they would isolate it, and they used the hemocytometer to kind of count each individual cell, and then they would assemble the DNA genome through PacBio and a lot of Illumina. That's going to be a common trend throughout this entire study. And then they also did phylogenetic studies, so that this was kind of a holistic study. So they used GenBank, and they were able to use, like ribosomes and DNA to figure out that this was in fact the protist that they were looking for, and it wasn't just some random like protozoan that they found. And to kind of continue with that, again, for the single-celled RNA sequencing, they used Illumina as well. For the 16S RNA sequencing, they took samples, from like a similar thing to when they did the protist taking, which was, at 0 days, 2 days, 7 days, and etc. And they also, one of the benefits of using Illumina are the amplicons that can be generated, so they took full advantage of that, and they, made amplicons, and that way they were able to do diversity and multivariate analyses on, like, again, bacteria that were able to, like, grow as a result of the presence of TIMU. In terms of metatranscriptomics, a bunch of sequel samples were collected at 2 and 28 days. We're going to see that there was a huge difference between the wild type and the mutant mouse, so that the 2 days and the 28 days was really, really important as like a time frame. And then they used Illumina again for base pairing. They also did PCR to just make stuff more abundant so they could see stuff. And they used RNA-FISH and RNA-scope as well. So without further ado, we're going to get into the actual research results of the study. I'm going to go ahead and turn my camera off, and then we can go talk about it. So the scientists quickly realized that there was a major rate difference in colonization. So essentially in the mutant mice, there'd be a huge explosion of protist content up until day 7 and it'd plateau until day 28, whereas in the wild type mouse, it'd be a gradual increase over that 28-day period. Importantly to note, by day 28, they were basically the same. It just was the method in which they got there. And the implication of that is there were major shifts in bacteria content, and a lot of those trends were basically analogous to how the colonization process works. So essentially, as the mutant type mice were getting colonized quicker, a lot of the changes of the bacteria content happened a little bit quicker as well, and then vice versa for the wild type mice. In both cases, though, you did see a major shift in bacteria content. Specifically in figure D, where we can see that purple bacteria, those are called firmicutes, and those are a gram-positive bacteria that are really prevalent in these mice. However, a lot of them you can see over that 28-day period kind of transition. You see some of this red actin-based bacteria, and that kind of just happens over that 28-day period. But perhaps the most interesting part is the congruence. So essentially from day 0 until day 28, there are a lot of differences in terms of how it happened because of the inherent difference between the wild type and the mutant mice. However, by day 28 and onwards, a lot of those changes actually went back to normal and are actually really similar to day 0, as we can see in figure 1D. Now, overall, they noticed 3 main trends, which are pretty much the only 3 that could exist. So essentially, some of the bacteria, like Bifidobacterium that we've covered in other lectures, and parabacteroids increased. Some of them, like the firmicutes, would end up decreasing, and then others would actually peak around day 21, and then they'd go down after that, which is also what we see in figure 1D, but we also see in 1G and 1H. So different bacteria behave the same way, but there was actually a major shift overall in the bacterial makeup of the microbiome after the inoculation of TIMU. And whether that was the wild type and whether that was the mutant type, there was a difference nonetheless. They just happened at different rates because remember, the colonization rate of those 2 were very different. Another thing that the researchers found was that there was a massive impact on metabolism depending on how much TIMU they found in the organism as well. So if you look at figures A, B, and C, they also kind of go more so along figure 1, and they kind of just show that there's a bacterial diversification, and then there's upregulation of a lot of different genes, and then they converge back to the same state by day 28. The really interesting stuff on this slide is actually figure 2D. So essentially, you can see that the presence of TIMU in both the wild type and the mutant genotypes show an upregulation of glycolysis, amino acid metabolism, peptidoglycan synthesis, and also the breakdown of sugars and fats. So essentially, it somehow caused the entire organism to work overtime in terms of all of its metabolic functions. And then we can see that there is that congruent shift from the firmicutes, the gram-positive bacteria, to a bunch of different new bacteria like bacteroids. And then that in turn increases chromosome segregation along with cell elongation and host immunity modulation, which also has major implications on the mouse's immune system. So phylogenetically, basically figures A through figure D all kind of show that they basically just assembled the DNA of the protist genome, and they were able to confirm that this was in fact TMU and that this is the one that they were looking for. And then from there, they were able to kind of further find out from the DNA that there was in fact even more metabolic increase. So we see, for example, like starch and sucrose metabolism, and along with like other glycan degradation, definitely increase, especially in the wild type. But it's important to note that up until now, all of these like metabolic increases are both in the wild type and the mutant, which is interesting because the next things that we're going to talk about are actually specifically for only the wild type, and that is these adhesins that kind of mediate virulence and cytoadherence, which is really, really big on how colonization happens and might have something to do with why the wild type actually has a very long and drawn out colonization, whereas the mutant instantly does it over 7 days. And we have these 4 distinct BSPA proteins, which essentially serve as extracellular immunogenic predictors, and they're upregulated only in the wild type mice. So essentially this suggests to us that B cell presence absolutely has a massive impact on TMU's TMU's effects, which is why that when we have the mutant gene that is B cell deficient, you don't see the adhesins and you don't see the 4 BSPA proteins either. So the next thing that the researchers wanted to figure out was exactly the life cycle of TMU. So the standard life cycle of a trichomonad has 3 stages: the trophozoite, the amoebid, and then the pseudocyst, which is basically a cyst that doesn't really have like the standard wall around it. And TMU is no different, but essentially trying to figure out like at what stages that they exist in, depending on what factors, was essentially the goal of this portion of the study. So through single cell profiling, they are able to basically form clusters, which basically exist when a certain cell is in a certain stage of their life cycle. So essentially they're able to find out all these different life cycles, and then really interesting, in 4D, they were able to find the clusters. So essentially, if you look at cluster 14, it is noticeably lower than all of the other clusters 1 through 15, and that's how they're able to basically determine that this cluster probably has lower transcriptional complexity, which means that it is probably the pseudocyst formation, which is not necessarily the final formation, but it's the one that is stationary. So there's probably going to be less activity going on and more so just like DNA replication and then just making that cyst a little bit bigger. So that's really interesting. And then I think perhaps most interestingly of all is going to be figures E and F, which basically shows that TMU has meiosis-specific genes. They're able to actually determine a meiosis phase along with a G1, S, and G2 phase. So this has crazy implications, again, for immunity, as this shows that, there's potential adaptation that is possible through sexual reproduction that would be otherwise impossible through things like binary fission that we normally associate with protists, and like things like bacteria. Staying on figure 4, we're basically going to look at only figure 4G now. If you notice, a lot of these terms should be pretty familiar, so especially the BSPA proteins, which are the immunoregulators, along with the adhesins, which are generally only found in those wild-type genotypes. Those actually show like a link with cluster 15. So essentially cluster 15 shows that even between the GF and conventionalized hosts, they both have these meiosis-based genes. So essentially the prediction right now is that the protist state, which of the 3 states the protist currently exists in, could mediate basically colonization rate and transmission. Just keep in mind, again, all of this stuff is generally only found in the wild-type genotype. So this might not apply to that B-cell deficient mutant, but this also is great information because now we know, it definitely is probably immunodependent in terms of how TMU acts based off of how it transfers from stage to stage. So going along with that life cycle theme, in figure 5, we basically find that certain genes using fish flow were able to be found as specific to certain states that TMU exists in when it's found in the lumen, but there also were universal genes. So essentially we could confirm that this was the correct protist, but at the same time we could figure out what state that they were existing in at that moment. And here comes the difference between the GE or conventionalized mice, is that depending on which one they were, certain clusters were actually found more often. So this was able to help confirm the cluster 14 as the one that exists as a pseudocyst, right? The one that had low transcriptional activity. And we can see it in figures G and H, how essentially depending on where you find them, the activity level differs, but it's actually not, quite as drastic as, for example, like figures F. And then as such, you were able to find out basically that cluster 14 is in fact the pseudocyst, which means that cluster 15 most likely is the meiosis gene as well. And then finally for figure 6, essentially we are talking about competition. So the researchers were able to find that TMU is both iron dependent and amino acid dependent. So it needs arginine and tryptophan as well. And conveniently, bacteria also need both of those things. So if you look at figures 6, C, and D, you can see that their activity actually goes up in an attempt to kind of like compete against the increased upregulation of things like ferredoxin and, the other taking of arginine and tryptophan in order to produce indole, which both bacteria and protists need. Now, interestingly enough, you can also see that in clusters 13 and 14 of figure 6b, you can see that the activity level goes down, and this is in congruence with the fact that this is most likely the pseudocyst stage, which we talked about in the last slide, where the life cycle stages are not all created equal in terms of activity, and you'd expect that that large mass of a pseudocyst would have less function, and as a result, less use of ferredoxin, and we do basically confirm that. So this pretty much confirmed to the researchers that stage 14, or at least cluster 14, is most likely the stage of the pseudocyst, and then stage 13 is most likely a transitional state. Perhaps most interestingly about this entire slide is the fact that this doesn't appear to be based off of B cell composition. So there's been other factors that are either determined on being germ-free or conventionalized, or wild type, or the mutant B cell deficient mice, and there'd be differences between the two. This is not one of those cases, so it basically suggests that there is direct competition between protists and bacteria, regardless of the mouse that was used. But with that being said, that was essentially the main, results findings. There were a couple more, but, as many as there were already, I felt like I was getting stretched thin. So I'm going to go ahead and turn on the camera now, and then we're going to talk about the discussion and move on. So there are a lot of takeaways from this. Most importantly, I guess, is the thing that the researchers were after, which is that TMU does, in fact, affect bacterial composition of the gut a lot. For example, like a lot of the gram-positive bacteria, like the firmicutes, turn into gram-negative bacteria, like the bacteroids. And we also know that the mutant B-cell deficient mice respond a lot quicker to the inoculation of TMU when compared to the wild-type mouse. We also know a lot about the life cycle in terms of the 3 phases that it'll shift through. And then we also know that, really interestingly, that the protist actually has meiosis genes. So essentially, there might be down the road some, leeway for figuring out, are they adaptable? Like, how do they develop resistance and stuff like that? And then we, of course, know that they compete at least with bacteria, which also, raises a lot of questions for how does it interact with fungi and other things in the gut. But overall, we know that this shows a lot of potential for commensal protists to be kind of used to induce certain immune responses, potentially in people one day, as we saw, it did over a large 28-day period in the mouse. Now, in terms of, concluding remarks, we know that, protist contribution to the microbiome might still even be like way more important than we initially thought. It can clearly cause a lot of different differences in like microbial content, different species of bacteria that we find, and different immune responses, and specifically a lot of metabolic functions as well. In terms of future, testing, I think ideally you can get it done in humans, but I do have a question on the next slide about like ethical implications, because I read through this and there's some stuff that I felt a little weird about. But also in humans, right, we have Trichomonas as a genus that's like a distant relative, so there could be a lot of important implications for diseases that might be caused with that strain. So overall, protists, they might be that next frontier that we need to like fully research to be able to find out exactly what their impact is overall. And that brings us to questions. So number one, what other factors could potentially affect protists like TMU? We talked a lot about just like the natural progression of stuff, but what if it wasn't controlled? Like, how would diet, antibiotic resistance, and other factors affect it? How might different immune cells other than B cells affect protists? So in this study, they had the mutant B, the B cell deficient, but like, what if there was another, deficiency in another different type of mutant mouse? How would that affect, or at least how would you predict that affects the colonization of Timu like at a certain rate. And then question 3, what ethical implications would prevent this kind of study from being replicated in humans? So I kind of have an idea in terms of like how they treated the mice and whatnot, but I'm really curious to think like that if that's a universal train of thought, or if you think that like you could probably do this in humans as well, as long as they were consenting. Question 4, how might Timu interaction with fungi differ from that of bacteria? So again, bacteria have been studied to death in terms of the microbiome. We're still, of course, learning all about it, a lot more all the time, but overall, we know a lot more about them. But in terms of like fungi as well, how do protist fungi interactions also shape the microbiome? And then last question, what environmental conditions might stimulate the shift between the life cycle? So we know the 3 phases of the life cycle now, but specifically, like, why do they shift between the 3? Why do they have 3? And then, like, are there external factors that would dictate that as well? And yeah, here are my references. Thank you so much for watching. I hope we all learned something new. And yeah, definitely ask any questions in the perusal, discussion below. So yeah, thanks y'all. See ya.